Yeah, right.
We good?
Hello, hello.
Not good.
Nope.
Hello. Well, that's not good. Hello. Hello. Well, there you go. That's weird.
Is it this one?
Yeah. Just two of us. Right.
Hello.
No. Hello. No. Hello. Hello. You want me to get that?
You may have to use the lapel. This usually works for the most part. They also said this works if there's a way to turn it on. Hello. So that's on. Hello. Still on. Hello. You can really keep it short. Hello. So keep it really short, well, especially since we're running out of time. Hello.
Does this work? This is not working either. Hello.
So that must mean it's gotta be something on here. The audio source. Which one is this? This is Lectern PC. Maybe. Hello. They're coming. Get out of the way before I break something. Okay.
But I think that
Turn this on. We couldn't figure out. This one.
Just turn the power button. You hold it down.
Oh, you hold it down. Okay.
Yeah.
Hello.
that's the one I
Oh. Now we have.
Hello. Okay, sorry for that technical difficulty. So today we have Dr. Buford from the Department of Medicine, the Division of Gerontology, Geriatrics, and Palliative Care. He's a professor there. Also a professor in the Department of Nutritional Sciences. He told me to keep this really short, so I'm going to skip through a lot of the things that I was going to mention. I will mention that he did his Ph.D. at Baylor after coming from Oklahoma, came to UAB in 2017. He's done a lot of things since then. We're really excited to have him. He's been on numerous grant peer review committees, serves as director of the UAB T32 pre and postdoctoral training program. A lot of his research and interviews have been in the Wall Street Journal, Reuters, Forbes Health, and Time Magazine. Today he's presenting Renin-Angiotensin system and age-related disabilities. So everybody give a hand for Dr. Buford. And I'm going to turn it over to him.
All right, thank you everyone for tolerating me while you have your lunch. So I appreciate that. So yeah, thank you to Dr. Wendy for inviting me. I got you back and you're present for us soon. So I'm gonna figure out getting used to this mic. It may take me a minute, so I apologize. But let's see if we are not. I may need the IT back. Let's see if that works. Fine. I don't know which one did that sound.
Testing. Yeah, that works too. So.
All right, there we go. We'll see how this goes. So again, thank you for the time. So I've spent the majority of my career focused on the problem of aging. And so here, I think the work that I'm going to do, or that I'm going to show, I hope shows a little bit of kind of just following where the science leads. I've kind of found myself getting into areas that, when we initially started out in this work years ago, never envisioned it would go this way. Hopefully also, a lot of the things we do, hopefully trying to cross paths with showing something that's relevant if you're a clinician or doing clinical research or doing basic science, hopefully the paths that, this has taken us, something is relevant. So the biggest problem and things that I talk about with people a lot are the aging population. And so I think at this point, most of the people are, you probably heard, you're aware of this. I'll just point out very quickly a couple of things. I have no idea which screen to look at, so you all help me. I'll pick over here, see if the pointer works. It's not seem to. We'll see here. So essentially, you can see the data on the left show that in basically one generation, and so these data are a little bit old now, they're from 2010, but the point being essentially in one generation, even if you exclude developing countries which have rapidly increased lifespan, look at places like North America, you've increased lifespan by about 10 years in just one single generation, and we expect that to continue. So as you can see on the changing graph on the right, I've always found this interesting to see it kind of graphically in real time, where you can see the population on the far left is children under the age of 5. And then on the far right, this is individuals over the age of 80, who in the next 10 years expected, if you slice those out by 5-year increments, will be the largest segment of the population. And so as we know, we've increased longevity, but along with longevity becomes increased risk of chronic and comorbid diseases. And so the work that I started out initially doing was really, my goal was to prevent, at the time, physical disability. It was really where I was focused in keeping people independent in their homes, out of nursing homes. And so if you're not familiar with this work, this is called the Nagy model, and it basically shows, appropriately enough, moving from the start of the model being on pathology, as you move toward functional limitation and disability. So it kind of disappears, I guess, when you hit the screen. So most of my work is focused in the area of functional limitations, where really we're working to prevent disability. And so some people consider it rehabilitation in the sense that you're trying to, work backward to remove the functional limitation. Others, consider it prevention research in the sense that you're, trying to prevent disability. Either interpretation is really correct. And so as we think about this, a lot of people, may not spend their time thinking about this. A lot of people we focus on our disease state of interest. But when you talk to older adults, the vast majority of them, the most important thing to them is staying independent in their home. And so they really value that more so than lifespan. And so a lot of times you'll have people who, the clinician working with them can understand the choices they make regarding their treatment because they're more concerned about quality of life than they are lifespan. And so I think intuitively we would understand that, as they become functionally disabled, they're less likely to be able to engage in their community. But it also clinically, this is where people start paying attention more, is there are very clear ways of identifying increased risks of hospitalizations and rehospitalizations, nursing home admissions, and higher rates of morbidity and mortality. So where if you look at the clinical setting, that's where these metrics that people start tracking are really important. And so just really quickly to give an example of that, I keep waiting for the day when I'm going to stop using this slide. It's pretty old now, but it gives the picture really well, where if you take a very simple measure of physical function, and so if we were to take people at the age of 65 and we measure gait speed, and so usually this is measured over a 4-meter course, sometimes 6-meter, but something that can be done in a clinic. So essentially you take somebody from here to the other side of the room and you just tell them to walk how they normally would. This is not, you're not asking them to push, you're just measuring their normal mobility. If you did that at age 65, and so between these two graphs for men and women, these are data from over 30,000 individuals, so it's pretty robust. And if you were to take their speed on that test, so these are the data for men. If you were to look at kind of the middle of the bottom tertile and the middle of the upper tertile, so these people have really high function, these people are not functioning so well at age 65, there's about a roughly a 17-year difference in life expectancy if you were to take that measure there as a prediction for their life expectancy. If you take the curve for women, the curves are exactly the same, shifted upward as you would expect for longer life expectancy. But what I would point out is that if you were kind of the middle of that upper tertile, you're pretty well functioning at age 65. Your median lifespan is actually 100 at this point already. So it's a pretty robust predictor of outcomes. It's associated, and these are just examples. This is repeated over and over in the literature that you can take these kind of functional measures and predict things like CVD, incident CVD, stroke, and then an area where I've been excited to see this, cardiac surgery was the first area. Other areas of surgery have now started to utilize this as a predictor of outcomes after surgery. And so one example here from cardiac surgery. So I'll just kind of give you for reference this gait speed measure. So 1 meter per second is generally considered kind of a clinically meaningful cut point where you fall above or below. Outcomes can change drastically. And that was long held before this study came out. And interestingly, post-operative mortality fell right along that line where your odds ratio significantly, was significantly higher if you went into surgery with poor function. And so accordingly, a few years later, the American Heart, American College of Cardiology, and American Geriatrics Society got together and pointed out that in these spaces, typically when we think about CVD or hypertension or anything in that space, we're looking at cardiovascular disease. And they got together and pointed out that we really don't have data on how to utilize interventions that are going to focus on these outcomes that are particularly important to older adults. And so an area which I've worked and focused on is the area of hypertension. And so depending on the day, the definition of hypertension bounces back and forth. But at the time, these data, no matter where the cut point is, you'll see that aging is obviously the biggest risk factor for hypertension. And so the kind of the intersection of aging and hypertension is where I spend a lot of my time. And so, again, this is just a very generic summary slide, but a lot of the time, I think we think about hypertension and we go to blood pressure and we think about cardiovascular disease. And that's, that's generally everyone's thoughts on it. But I developed this figure a while back just to point out yes, we have the issues with CVD, but we also have significantly greater risk of cognitive decline, functional decline, and falls and fractures that ultimately contribute to those things, all as a kind of compilation of both aging together with hypertension. And so these are just one example of data. These are 3 different scales where you can look at physical disability risk, and the incidence goes up as you move from, in particular, uncontrolled hypertension increases risk. And similarly, these were actually data from our group, where if you tracked over time, just over a 1-year period, people who are normotensive or people who are hypertensive, older adults, their gait speed, this is on a slightly longer walking task, but again, the normotensive individuals not only started in a better place, they basically maintained it. And in a 1-year time, you may not have a reference for this, but a decline of about 0.05 meters per second is clinically significant. And so in a 1-year period, hypertensive individuals declined at that rate. And so again, I'll kind of move past these, but same kind of story where you see with cognitive change, as you move from pre-hypertension to hypertension, and particularly to uncontrolled hypertension, your risk of cognitive decline, as well as MCI and dementia increases substantially. And so the work, as I began, working on the renin-angiotensin system again. And I was thinking about this in a very, very different way than nearly anyone, I worked with, because I wasn't really thinking about it from a blood pressure perspective. I've put here very intentionally a very, very simplistic model of the kind of our initial old understanding, is what I'll call it, of the renin-angiotensin system. How many people, if they're not really familiar with it, might still think of it, where we think of really trying to either block the AT1 receptor with ARBs, or block the conversion of angiotensin 1 to 2 with ACE inhibitors. I put this here to kind of set where the story began in this space. And so some of the earliest data, I think, that contributed to kind of this change in direction and understanding of the impact was from Patrick de la Fontaine's group way back in 2001. And so these were actually data taken from skeletal muscle of rodents, where they showed that, so they were really looking at IGF-1. I won't really get into that, if you're not a muscle person, but the point being, gastrocnemius weight, so muscle size, we'll simplify it there, was way lower when animals were provided angiotensin 2. So it was kind of one of the earliest links to understanding, well, how could something impact, outside the circulatory system, outside the systemic, just control of blood pressure. And I think as it relates to physical disability in particular, skeletal muscle is one of the biggest drivers. And so, if you love biochemistry, I'd love to talk things like this with you. I'm not going to harbor on it for those of you who don't, but the point being is this was a review from members of de la Fontaine's group, pointing out just different ways within skeletal muscle, different pathways in which angiotensin 2 could act to influence either protein synthesis or degradation. And subsequently, other mechanisms have been shown indicating that there's actually an independent mitochondrial renin-angiotensin system. There's also a renin-angiotensin system in myoblasts, which is responsive to, in this case, they say stretch, but essentially if you think about even like resistance exercise, impacting local renin-angiotensin systems. And so subsequently, there was a group who did some work to look at epidemiologic cohorts. I think my thinking is they found interest in that space. And so I'm going to try to, I'm not sure if get that out of the way, where the best place is. But so these data were taken from, I believe, the Women's Health Studies. So it was over 3,000 women. And they categorized people based upon their antihypertensive medication use. And they were followed for about 3 years. And, I realize the lines are tough to distinguish, but they were categorized as either individuals who had continuously taken ACE inhibitors the entire period, who had intermittently taken them, so they may have changed, gone on and off, people who had continuously taken taken other antihypertensive drugs that didn't include ACE inhibitors, and people who were normotensive had never taken any of these drugs. And the point being is one of those lines is clearly separated from the others. This is muscle strength, and this is a gait speed measure. And individuals who took ACE inhibitors continuously seem to have a sustained level of function compared to the others. And so it's a very interesting epidemiologic finding. What it meant was still kind of to be played out. And so from there, several people did trials in this, thinking, oh, can we just give people ACE inhibitors instead of the other meds? The answer was no, that wasn't the case. And so kind of died on the vine in that setting. Let me see. Well, listen, there we go. So subsequently, one of my good friends and colleagues, Dr. Christy Carter, who's now at the NIH, started doing some work in her rodents, and we wondered really, was this an effect not of the medication, but maybe some kind of interaction between the medication, and were those people just more physically active, perhaps, and was that explaining that? And so she did a study. These are from Fisher 344 brown Norway rats. Very simple study just to give them enalapril or exercise. It was short term. I don't remember in this particular study the exact duration, but it was a pretty short term study. And they just looked at treadmill endurance just to see after the fact, were they going to be able to increase, this is similar to a measure of cardiopulmonary endurance in humans. And so effects were at least in that case additive, if not synergistic. And so that was kind of the first step to say, okay, maybe there's some kind of interaction with exercise going on. So subsequently, I did a retrospective analysis then. So taking that work from animals, and then we started looking at a retrospective analysis of a clinical trial in humans. And so in this study, people were given exercise. I believe this one was from a study of about 400 individuals, and they were given exercise, but their medications were not necessarily randomized. So it was whatever they were taking clinically, but they were given exercise. And we wanted to go back, kind of do what that other study had done, and look at the different types of medications and see if it mattered. And so what we saw in that study, again, was the individuals who we categorized as ACE inhibitor users had, so this is that gait speed measure. This is another, it's called the short physical performance battery, another measure of function for older adults. And what we saw was, this is a clinically meaningful cut point right between here and here. And then same thing here, right above 1.0 on this. And essentially no effect in the others. And so that had interest to me, both from the perspective of the antihypertensive medication mattering, but as well as the fact that all of these individuals in these two groups did a lot of exercise and didn't get much out of it. Again, it wasn't randomized, can't prove anything, but that's kind of like sparked my interest. So then I would get the question about these two groups in blue. What about the people who did not exercise? So what if they just took an ACE inhibitor and did not exercise? So we looked at that. And so you can see the bars in blue. These are all ACE inhibitor users. If they exercised, here was their change in those two scores. If they did not exercise, here was their change in their two scores. So again, kind of showing that it wasn't just giving that drug and then expecting that they improve, that there seemed to be some kind of interaction between the two. Yeah, so if you don't work in this space, I apologize for this hazard plot, but the point being here, you could look at two groups, one group in blue, one group in red, who either got essentially a health education, a control intervention, or people who got exercise. And what you're trying to see is within either ACE inhibitor users here, other antihypertensive users here, or normotensive, non-antihypertensive users here, the spread between those two groups to show the effect that the exercise had. And so here you can see you had a meaningful, statistically significant difference in ACE inhibitor users, where the exercise intervention was apparently more effective in those individuals than it was in those other groups. In particular, the normotensive individuals, who you'd think would be the healthiest, who might, so these were not healthy people by any means. They had to have functional deficits when the study started, but they did not really get much out of that. And so this is over a 3-year period. So then you have people who spent 3 years exercising, and then you wonder why people get mad about exercise if they don't see a benefit. So, I was as intrigued about these groups too, about how do we improve that. So again, kind of there's this renin-angiotensin thing and the medications that I'm interested in, as well as, well, why are people not responding to the exercise, and how do we get them there? So I took several slides out of here for time, but just to say, this was kind of our conceptual model. Showing we had evidence showing some potential improvements with ACE inhibitors and exercise together on things like blood glucose, IL-6 and adiponectin, and then some skeletal muscle adaptations. So we think there's some things going on systemically and something specific to skeletal muscle in particular, likely trending toward the kind of fiber type shifts, capillarization. We also saw some changes in kind of fat utilization within the tissues. And so from there, we utilized this data that we had, and we proposed a pilot trial, because the argument up to this point would have been, well, you still haven't randomized medications. We still have no idea. It's, there's a huge mix of types and what their prescription is. So we got funding from the American Heart Association to do a pilot. I apologize, it probably looks larger on the screen than it does for you all, but Point being, we did a 24-week trial where we randomized people to one of 3 first-line antihypertensive medications, so perindopril, losartan, or HCTZ. I can go into afterward, if you like, the reasons for choosing those. But essentially, we wanted 2 things that affected the renin-angiotensin system and one that did not, looking at those together. And then there was also at the time, and still is, data to suggest which medication to choose may depend on what mode of exercise you choose. So whether you do aerobic exercise or resistance exercise, the type of medication you're using may actually influence your results. So we wanted to do that in our pilot study, and we did. And it did turn out that it actually did seem to matter. And so out of a number of graphs I picked here, if you had resistance and aerobic exercise together, there were weren't really sure what to make of that. With aerobic exercise only, this is what we hypothesized, it was pretty clear that there appeared to be kind of a step down approach in this, this gait speed response. So again, this was not powered in any way to detect significance. It was just enough to, can we tell NIH, hey, we want to investigate this further? Luckily, we were successful on that. And despite COVID, we are doing our best. We're in year 6 of the study now. So, that was, the most fun, I think, of COVID was if you were doing an exercise trial, which is all our center does. Everybody was just under the assumption, that we were just going to be making people spew more virus. And so it's going to be very dangerous. And so very proud to say, like, our team was able to continue these studies safely. That being said, still a big delay compared to what we had anticipated, but we are close here. So in this case, we have currently a 3-site trial where we're doing actually 32 weeks of study, but we have a period before we exercise to see just what happens when you just put people and get them stable on medications, and then we have a period after exercise to kind of look at detraining. And so then I think about 5 months in between where they're actually exercising and on their prescribed meds. So like I said, we are probably about a year out maybe from having starting to get to analyze some results from that study. And so, I just kind of summarized here. I've always found this, I don't know, I guess maybe pride myself on the fact that we were able to kind of take things from what we saw in epidemiology, utilize animals in a way that got us, back to humans. And so that's the way we try to think of things, is being able to move things that direction. So then, that's kind of the end right there, as we're still working on that trial, but we still had questions we wanted to ask related to the kind of newer, more newly discovered arm of the renin-angiotensin system. And so I put here connections to cognitive function via the gut microbiome, because this was not an area that we hypothesized at all. This happened because we were still kind of continuing to investigate physical function, and that's where the data took us. So subsequently, we had been exploring FDA-approved medications here, but we had more questions about this arm of the renin-angiotensin system. So it's not new by any means, but newer, more recent than kind of the canonical axis. And so this arm of the system actually works in a beneficial manner via the effects primarily of angiotensin 1-7. So essentially, Ang1 can get cleaved. Instead of to Ang2, we become Ang1-7, bind via this MOS axis and have beneficial effects. And so we wanted to ask questions about, instead of just, what if we could just not block this side of the arm, but what if we could actually induce this side of the arm? And so there were no FDA-approved medications. There was no way to do that in humans. And so we wanted to ask those questions. And so we thought, well, let's go back to our animal model. And so we started initially with what's called here an ACE2 activator. Dizzy is the short name for it. And so this is this was a colleague of ours when I was at the University of Florida was working with this. It's actually used to treat African sleeping sickness. It was very interesting for me. And so we utilized it in a couple of different ways, but one was, so we did this study looking at essentially a month long and saw a tremendous decrease in body weight. So these are those F344 brown Norway rats, which are a great model of aging, and they get huge. I mean, so if you work with mice, I mean, we're essentially working with guinea pigs in this case. I mean, they are massive. And so we saw a huge decrease in body weight, which could be very traumatic in an aging sense. And so, but we also looked at, sorry, it's going to be a constant kind of shell game moving those around. We also looked at kind of a muscle, we looked at muscle and adipose tissue. We want to make sure we're not making them lose more muscle. Hopefully it's in the adipose tissue space, which we had seen a little bit of earlier, and that was the case. And so this is kind of the fat to lean ratio. Up here is muscle, which you can see was essentially unchanged, and then the white adipose tissue. So, I mean, that was beneficial. The problem is Dizzy is extremely toxic, like it's given for African sleeping sickness. We're not probably going to be delivering it to patients regularly. So we had to come up with a different plan for how are we going to activate ACE2. And so the initial strategy we utilized, so this was Dr. Mohan Rosado was our colleague, and so they were actually creating Ang2 in plants. So it was actually in plant chloroplasts, and then you would crush it up, give it to the animals, and then actually it was in their food. What we discovered when we actually wanted to do a study, the problem is plants grow really slowly. It's really hard to produce a lot of that on a large scale for a big project. And so this is kind of where, you know, I'd say for me, it was very interesting to think, you know, I'm kind of a muscle person by training. And so there was this link that we'd seen with the gut microbiome. And so ACE2, which again, I guess for everybody over the past 2 or 3 years, ACE2 was kind of noted because that was the receptor that COVID got in the body by. So at the time, this was also an issue we were concerned about, was where our study is going to get shut down because they were investigating ACE2. But we see that this is actually active within the gut microbiota. And so this title here is Defective Intestinal Amino Acid Absorption in ACE2 null mice. Looking back now, I didn't catch at the time. Subsequently, it's really critical for the metabolism of tryptophan. And that kind of comes into play later, and we figured that out. But essentially, yeah, you have a very difficult, I don't know if it completely inhibits the metabolism of tryptophan, but it has a huge role. So then they started thinking about, well, how can we utilize the gut microbiome? And so the strategy we thought about then for delivering this was through a genetically modified probiotic. And so now we have a colleague, Dr. Lee at UF, who creates these genetically modified probiotics that then express the protein of interest, in particular, Ang17 or ACE2. We primarily work with Ang17 now. So you can essentially gavage it to the animal, and then everyone always asks me, it does survive, it survives the gut, and then expresses in the circulation. And so we're going to, I never do this. We're going to take a shot, see if it works. If not, we won't spend any time on it. All right. Who's got a guess at which one's going to work? All right. So essentially, so these are the animals that we utilize. So this is a colleague of ours, just kind of showing an example. So this was published. This is actually alongside one of our articles. So you take this. That was also a question we get from IACUC, is whether these older animals could actually handle that gavage. They handle it no problem, but you do have to force feed them. So, all right. Now see if we can get back to the slides. There we go. So we did a proof of concept study. We just wanted to basically start to investigate, is there anything going on if we give these? And so we did some behavioral testing. We did some functional testing. And one of the most interesting things that we found, we gave this for 3 months, and we saw this increase in novel object time. So this is a behavioral task that looks at object recognition. So it's kind of a very blunt task, I guess I would say, of memory or recognition memory. And so we saw it was a smaller study, but interesting effects enough, particularly if you're familiar with the NIH and NIA right now, Alzheimer's and cognitive function, is really where, the interest is. And so we were like, okay, well, this is very interesting. So we wanted to explore a bit further. And so as we dug into the literature again, it wasn't just the gut where ACE2 was playing a key role. There's actually a well-established literature of how the renin-angiotensin system and particularly Ang-1-7 impact the brain and the central nervous system. Again, some of those similar pathways shared with skeletal muscle. And so we looked at it, and the first thing we had to know is, again, is it going to survive the gut? Is it going to do anything? And so this was from that gavage. And so this is circulating Ang-1-7, and we always kind of benchmark it to Ang-1-2. And then we looked at dosing, whether they didn't get it at all, or they got it 1, 3, or 7 times a week. And so 3 times a week actually turns out to be the optimal, but the primary point was that we could deliver this via orally and actually get the effect we were looking for. Reason that was important, because there are actually studies going on. There's a couple of cancer trials. I believe there's some people doing it with cognitive function, but they have to deliver either repeated injections or IVs. There's no way to deliver this orally at this point. And so then again, so by its nature, we also started getting into work in the gut microbiome, which is now everywhere. But at the time, if you'd have told me moving from muscle to working with microbiota, and I would have laughed. But similarly, so these are some data showing measures of richness, which is just saying how many species are actually in your gut. And I think these are actually from, if I recall right, stool samples specifically. But again, they kind of lined up with the dosing and what was optimal. 3 times a week across all the things did tend to pop out as the optimal setup. And so other key thing we really had to show was that we utilized the lactobacillus backbone for this. And so if we weren't detecting the lactobacillus actually in the microbiota, we'd have really gotten a lot of questions. And you can see, essentially, I mean, it wasn't showing up at all. This particular lactobacilli wasn't showing up at all unless we actually dosed it. And then we saw some other interesting findings, particularly for this bug Akkermansia, which has been shown across a variety of conditions to be associated, in particular with inflammatory conditions, by and large. So you can see atherosclerosis, obesity, et cetera. And so then, started trying to think about that communication. So if we have some kind of communication going on from the gut to the brain, again, so this is kind of an area where now I know a lot more people are working. It's a big pool to choose from. It's like, well, how do I choose? And so as I kind of did my homework and kind of developed this figure, tryptophan metabolism jumped out at me. And so as I mentioned, why tryptophan jumped out to me is because that connection with ACE2 in the gut. And so we started looking at some of these measures. So even doing this figure bored me, so I don't want to dig into it with all of you. I had to show this for reviewers. But point being, when tryptophan gets metabolized, it gets broken down into a variety of things. And depending on it, just like the renin-angiotensin system, depending on how it's cleaved and what's produced, it can either be neuroprotective or it can be neurotoxic. And what all those factors are that influence those cleavage points, we don't really understand well at this point. But again, so we were kind of interested in the brain. We'd had enough to say, all right, there's something going on. So again, so this was it was just a 4-week dosing study. And so we just kind of did a very quick scan of some neuroinflammatory genes and looked at gene expression in the prefrontal cortex. And so what we saw, again, there were a few others, but pretty consistently that 3 times a week dosing seemed to reduce, this is just gene expression again, a select number of genes in the prefrontal cortex. And so again, so there we were just, it's not anything definitive, but enough that we were like, okay, we want to keep going further. So then another question we typically get was, how does this compare to if you just deliver Ang-1-7 systemically? So I said that you can give it through injections, like it's available. So we gave it in the circulation. And so we would just give subcutaneous injections. And we did see substantial increases in circulating Ang-1-7, just like we did with the other. But as you would expect, there wasn't any change in the microbiome because we hadn't done anything to modify that. There were no changes in tryptophan metabolism, which probably makes sense based on what I've said so far. And then there were no differences in the prefrontal cortex or other tissues that we had seen things in. So there was a distinct difference between combining kind of those potential benefits of the Ang-1-7 with the microbiome, as well as the benefit of getting to give it orally. And so, yeah, so then I kind of summarize it again saying, you know, we really kind of pride ourselves again on kind of reverse translating, saying like, okay, yes, this model, but not just you focusing on the model, but focusing on the question. And so we've done studies now where we've given ACE2 or Ang-1-7 and continuing the ultimate goal, which we're not there yet, but we work with Dr. Lee. The goal is still to figure out how can we get this back to humans. And so that'll be interesting to see if we are able to do that. As I understand it from Dr. Lee, the biggest problem in that step is they really look at, are you certain that you have all the antibiotic resistant genes out of those constructs before we can be able to give them to humans? And so luckily that's outside my wheelhouse and she's working on that. And so we'll see how it goes. And so just wanted to finish by kind of saying where those things have taken us and kind of some of the more recent work and future directions that we're looking at. And, you know, highlight some of the things. Again, these are not areas in, I guess the exercise interaction is an area we hypothesized, but now we're kind of looking at, can this work in Alzheimer's disease more specifically, not just cognition, in healthy individuals, as well as really seeing some sex differences in all of this. And so this was work done by a colleague at Samford, who postdoc here, Dr. Jumbo Luciani. And I'm going to do my best to describe this, but essentially, so this is a fly model of Alzheimer's disease. Well, we actually used a fly model of aging and a fly model of Alzheimer's disease. And so, so you take these basically photon tubes and put flies in, and flies are naturally attracted to light. So if you put the flies on two sides, you got a dark side and a light side, they're always going to gravitate toward the light. So what she'll do is put and I can't describe, I'm not exactly sure what the smell is, but you put a really nasty smell that any smart fly would really hate so much. They're like, I'm going to go into the dark side because it's not worth it. And so if you train normal flies, like they can figure it out pretty well. Okay. But she also has a model of Alzheimer's disease, which cannot really learn that. They just continue to, and so she has to do these tests to say it's not just a smell issue. It's not because they can't smell. So obviously you have to do that. But it's a little bit of a messy graph. But the point being here, these are females and these are males here. And so just in summary across the groups, so they were untrained. You know, they didn't go to the dark because that's not what they liked. So this is just kind of the basal condition. So in after training in females, both the AD flies and the male and the aged model flies figured it out. And so this maybe is a comment on sex differences in men and women, but the males did not do that, let's say. So this is the control fly, just kind of the aging model. In males, they figured it out. The Alzheimer's model, nothing. Like they just couldn't process it. And so then you gave them this is the LPA. So this is our probiotic. Here, it did not really benefit the females, interestingly. So particularly the AD females. The males, so again, in just the age condition, this is essentially the effect that we saw with just the training. But the LPA did seem to have some kind of an effect in the male Alzheimer's flies. And so there's a number of different kind of interesting questions that I can see coming from this. But first off, the renin-angiotensin system components throughout various regions of the body differ in expression by sex, and they also differ. The rates of Alzheimer's disease obviously also differ significantly by gender. And so I think there's a variety of ways to take this. And so sex differences is one of the things that we really want to kind of continue to investigate further. And so you see that kind of persist a little bit. So a study, this is from Dr. Abby Hernandez. So she's a postdoc in our group, hopefully will be on faculty soon, where it looked at just the effects of the exercise on just that exhaustion time again. So kind of cardiopulmonary fitness. And you see in males and females, kind of obviously the exercise had impact like that. That's not really a question. But here is the control versus just a null probiotic versus the genetically modified. And you can see kind of a step increase across the exercise group in the females. So then we looked at kind of two measures of cognition or domains of cognition. If you're a neuroscientist, please don't shoot me. But this is kind of a measure of anxiety. And so you can see in the males here, interestingly, despite this, these don't necessarily line up perfect with the exhaustion time. But in the exercise group, these weren't, based on our numbers, weren't significant. But you can see kind of the impact of the exercise with the different probiotics and kind of a nice step approach. Here in the females, didn't really impact anxiety-like behaviors. And so then if you look at novel object recognition, so recognition memory seemed to be affected in the females, but not in the males. And so different domains of cognition or cognition-like abilities seem to differ, but there seems to be kind of a consistent pattern of these compounds differ whether you give them in a sedentary state or an exercise trained state. And so this is just kind of the last data I'll show, which just to kind of show the work we've been doing. So we're getting into data that are far above my pay grade at this point. And so Dr. Liliana Baptista has been a postdoc and now a longtime collaborator working with us. And so she's done some multi-omic analyses where taking animals who either received those different compounds and they exercised or they didn't, and then looked across a variety of tissues, and then figuring out how to actually compile data from across those tissues from different levels. So from RNA-seq to metabolomic analyses. And so this is a study, hopefully is about to be published, where we saw, again, so this is just microbiome data, just summaries saying it looks like the LPA and the more so than the just the null probiotic seems to affect the microbiome, which is interesting because the backbone of those two things is the same. And there may or may not be differences in exercise, and it may or may not differ by sex. So these are smaller sample sizes, so we still have a little more to figure out here. But the cool part was when she started putting some of these graphs together for me, I mean, she was going to have to handhold me, but so she took data from across a variety of tissues. And so these were data from the prefrontal cortex. And when you're looking at RNA-seq data combined with the microbiome data, combined with metabolomic data. And the cool thing to me of what I could pull from it was really that, again, we saw tryptophan, picolinic acid. So these were some of those measures that we saw in some of those pathways that I worked on, showing up when you looked at across a variety of conditions, across a variety of pathway or even tissue types. And so she's continuing work in this area. Very similar findings we saw in the liver. So again, I didn't really give a whole lot of a liver story here, but the liver is clearly integrated with all of these things. And you see kynurenine and those tryptophan pathways show up again. So I'm going to leave it there. I'll be happy to take any questions. We just received an R21 to look at delivery of this compound in Alzheimer's model of rat. We're working with Dr. Lori McMahon, who's now, many of you may know, she's now at MUSC. as well as following up on kind of those sex differences, effects, and really going deeper into kind of behavioral and cognitive profiling from compounds. So, again, I'm going to kind of just put these up here, but to summarize, I think for me, it's just been very interesting to understand and get to go across the different, different types of animals, different types of research models, different, going from skeletal muscle to the gut microbiome, to cognition, to the whole person. And so it's been just kind of a fun journey for me. So I appreciate everybody, Adam, for inviting me and allowing me to share a little bit about it. So with that, I will just thank all the collaborators and funding agencies. So thank you. Yeah, Rakesh.
Thanks. Any thought about, or in your experiments, was the diet controlled, across some of these interventions? And if so, what was it controlled?
Diet's always, so our diet's always controlled. So that's one of the things that the technicians have to do daily is monitor. I mean, they have to monitor body weight, they have to monitor food intake. So Christy Carter is always, like, she's done this for a long time. And so food intake was one of her major things we focus on. So it's not only are they on the same diet, we actually monitor food intake to understand, are they intaking more or less? Is that influencing it? The answer to that is no, it's not. Now, Dr. Hernandez, who I hope will join us soon. So she's done a lot of work with different types of diets. So with caloric restriction or with ketogenic diets, things like that. So we have not varied the diet. That's a lot of things to vary the compounds, to vary the exercise, then start varying the diet. We have not done that yet. So it's always been a standard chow diet. Yeah. Or just somebody else is going to have to pick hands this time.
So do you think there's a concentration of an ACE inhibitor you could use in a non-hypertensive person that would work?
Yeah. So I'm not a clinician, so I'm gonna be real careful there to caveat. In some contexts, so if you, we'll step over to, like, let's say statins right now, right? You could ask a lot of people and they would say everybody needs to take a statin. I don't necessarily I don't necessarily agree with that, but there's people who make that argument. I haven't seen that argument here. Is it possible? Yeah, I think it's possible that there might be a low dose that would be impactful. In this, just in the context that I'm talking about, it does seem that if you're sedentary, it may be a moot point. Maybe you have to be exercising. The way I give that, a lot of the work that I do revolves on exercise. I say you can't take anabolic steroids, go sit on the couch and then expect to get huge. And because a lot of physiologic effects or off-target effects of drugs change when the cellular energy environment changes. So depending on if there's an energy need or not. So I think the likely answer is yes. Do I think it's likely to happen? No. And I will say in the case of things like statins or, metformin, and metformin is one that a lot of people say, oh, everybody needs to take metformin. Well, it's the opposite. There are several studies that show when they exercise, the impacts of exercise are blunted by metformin, both in resistance exercise and aerobic exercise. So if you're sedentary, maybe it's beneficial. If you're active, it may actually be harmful. I mean, I don't think harmful is the right word, but so possibly, I guess, a long-winded way of saying.
So on that same sort of line of thinking, is there a certain level of exercise that you have to get past for, let's say, a 65-year-old individual? Is there a certain amount that you have to get past before it has a benefit?
Yeah, I'm very careful about that question. I don't think the answer, I think the answer to that is no. Your level of benefit is going to increase in a dose responsive manner. The more that you do in this case, caveat, there are people who can do too much exercise. There are very few of us who are those people. So for the vast majority of people, it's a dose response that, like, the more that you do. But also we know a lot about very low, let's say for older individuals, lower levels of activity can be very beneficial in a variety of ways. And particularly in intensity, things like that. There's, the 10,000 steps per day that was an arbitrary thing that probably was never realistic. Now there's data showing that somewhere in the 7,000 to 8,000 steps per day can be really highly protective of cardiovascular events and mortality. And so that messaging is very different for me, and to say that like any is better than none. So, yeah.
Going back to your 2020 paper with some of the initial probiotic, you showed a lot of suppression of pro-inflammatory. Is there also an induction of scavengers like catalase or TOD or anything?
Oh, I would love to collaborate with somebody who wants to take some tissues and analyze. I'd love it. No, we haven't done it. We haven't done any of that.
Similar to that question, you showed the cytokines are decreased. Is it really beneficial?
Gene expression, just to clarify, yeah.
Beneficial to decrease them from the baseline, or is there any evidence that they shouldn't be there?
But yeah, again, so I'm gonna go back to the fact that I'm not a neuroscientist, but my understanding from the colleagues that I work with in that particular model is that reducing neuroinflammatory analytes would be a benefit. But I'm going to caveat that again to say that is not exactly my expertise. So I think there's still room to investigate that. But there was nothing else, I guess I'll say, that looked like what we were doing was a negative to the animals. Everything else seemed to be benefiting them, aligned with their cognitive changes.
Maybe out of box question, some of the large animals hibernate. What is the angiotensin axis there?
In hibernation?
Yeah.
I have no earthly idea. I do have a colleague though, who, I mean, in general, works with him, who focuses in comparative models. And so Dr. Ostad may have a good answer for that, but he doesn't really work in the renin-angiotensin system. So I don't know if he'd know that answer either, but I mean, I guess I don't have a great feeling for that. One way of thinking about that for humans would be that essentially we go in hibernation every night. So you could look at diurnal changes in some of these things and potentially get some kind of insight. I'm trying to speculate to get to a bear right now, but I don't know. I don't know.
I may have misunderstood this. How do you measure anxiety in a small animal? Is this a chemical measurement? Or?
No, so it's essentially a let's see here. It's a maze where I don't think I have a picture of it. It's called a T maze, and so essentially there is a portion of the maze that has sides around it, so it makes them feel secure, right? And then there's a portion where they can venture out, and basically, essentially, they could see the ground, right? So you can think about, if you're an animal, thinking about falling, like that would induce anxiety. The more time that they spend hidden in the box or in the end where they have the walls, they're thought to have a greater level of anxiety. So again, I'm also going to hinge on, like, I didn't create that task. I can't go into the data that verifies, but I think it is pretty widely accepted as kind of a measure of anxiety in animals. Yeah. Those were all here before we started. So.
If there's nothing further, everyone join me.
Yeah, thank you. Appreciate it. Stop.
Go ahead, you can mute us or end it.